In this lesson, you will learn about the review of CMC for biotechnology-derived products in Japan. Please refer to the video titled Review of CMC for basic information related to the quality review of drugs in general. Biotechnology-derived products are products that contain proteins or peptides manufactured by utilizing recombinant DNA technology or cell culture technology. Bacteria such as E. coli, yeast, and animal cells are used for protein biosynthesis in the manufacturing process. These recombinant proteins and peptides can be highly purified and well characterized using an appropriate set of analytical procedures. Typical examples of biotechnology-derived products include enzymes, hormones, cytokines, and antibodies. Biotechnology-derived products have much larger molecular weights and more complex structures than small molecule drugs. Because the products are manufactured using the biosynthetic processes of living organisms, the active ingredients of these products generally exhibit heterogeneity associated with post-translational modifications such as glycosylation. Since biotechnology-derived products are manufactured using genetically modified cells, product-derived variants may occur during the manufacturing process and storage. The variants and residual raw materials used in the manufacturing process may become impurities and affect the efficacy and safety of the product. In addition, the use of raw materials of biological origin carries the risk of contamination with viruses and bacteria. These quality attributes may vary depending on various factors in the manufacturing process. In the quality review, it is necessary to ensure that biotechnology-derived products comply with Japanese regulations such as the Pmda Act and the Japanese Pharmacopoeia, as well as chemically synthesized drugs. It is also important to ensure that products are consistently manufactured with confirmed efficacy and safety by evaluating that the manufacturing process is well controlled and that specifications are adequately defined. In addition, for biotechnology-derived products, it is important to ensure that risks from adventitious agents are adequately controlled and that quality control is performed considering the characteristics of recombinant proteins as described above. From the perspective of ensuring patient safety, it is particularly important to ensure safety against adventitious agents. In Japan, when raw materials of human or animal origin, such as serum and enzymes, are used, it is necessary to confirm that they comply with the standards for biological raw materials. It is required that raw materials contaminated with bacteria, fungi, viruses, etc., are excluded by screening tests upon the acceptance, that inactivation and removal processes are performed in the manufacturing process unless there is a valid reason not to do so, and that records are kept at the time of acceptance. In addition, complementary approaches are recommended in ICH Q5A as reasonable measures to prevent viral contamination and ensure safety. To this end, it is important to select and test raw materials, including cell lines and other components of the culture media, to exclude viruses that may be infectious or pathogenic to humans. Assess the capacity of the manufacturing process to inactivate and remove infectious viruses, and test the product for infectious viruses at appropriate steps of the manufacturing process. When starting a clinical trial for investigational products containing recombinant protein as an active ingredient, it is necessary to attach a document that contains at least the following information to the clinical trial notification. 1. The process flow diagram of the test drug. 2. that the cell bank is not contaminated with infectious agents. 3. That the unprocessed bulk medium is not contaminated with infectious agents. 4. Compliance with the standards for biological raw materials, if raw materials of human or animal origin are used. 5. Viral safety of the investigational products. And 6. Removal status of impurities, as well as provisional specifications for safety among the specification tests for the drug substance and drug product. For the concept of safety assurance in the first-in-human studies, please refer to the guidance for establishing safety in first-in-human studies during drug development.